Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at how to use genetic engineering to make a transgenic organism. Before we get going, I'd just like to recap what a gene is. A gene is a section of DNA that codes for the production of a protein. Now this protein might be a structural protein like collagen or keratin. It could be an enzyme that controls chemical reactions in a cell. It could be a hormone like insulin, or it could be a functional protein like antibodies or hemoglobin in a red blood cell. So if we're going to make a transgenic organism, we need to understand what genetic modification and recombinant DNA is. So genetic modification is when a gene is cut out of the DNA of one species and inserted into the DNA of another species. The new DNA is called recombinant because it's DNA made from two different organisms that have been cut and recombined together. Now, a transgenic organism is an organism that has recombinant genes for DNA from two different species. A transgenic organism will be able to make the protein that the new gene codes for. For example, transgenic bacteria can produce human insulin. We've got some examples of transgenic organisms on this page, and you'll notice that a number of them glow. And this is because they have had a gene from a particular species of jellyfish inserted into their chromosomes, into their genotype, so that they express that gene and they are glowing. This is an easy way for scientists to be able to see whether or not a gene has been successfully taken up by the organism you wish to insert the gene into. So how exactly can we do this? Well, let's have a look at this classic exam question here. Explain how genetic engineering can produce transgenic bacteria that can make human proteins. Well, the diagram below explains exactly what's going on. Firstly, we have to take the human cell, and in this case we're going to be making the human protein insulin. So a specific enzyme is used to cut that target gene from the human DNA. And at the same time, little rings or plasmids from a bacteria are isolated, and the same specific enzyme is used to cut open the plasmid DNA. So we have our insulin gene, our target gene from the human chromosome, and we have the bacterial plasmid that have both been cut open. The open plasmid and the target genes are mixed together with a different enzyme called DNA ligase, and the DNA ligase will join together the target gene and the DNA plasmid to form a recombinant plasmid. The recombinant plasmid can then be inserted into bacterial cells, and the bacterial cells are then allowed to multiply. Firstly in a lab and secondly in a fermenter. And the human gene can then be switched on, and the bacterial cells containing the recombinant plasmids will start to produce the human protein. And we can do this on a huge scale within those fermenters, an industrial scale, and we can then produce, in this case, the insulin protein, the human insulin protein. This process can also be done with many other proteins, so long as we can identify the gene that codes for that specific protein. So I'll run through that again, and this will be a 6-mark exam answer. Specific enzymes are used to cut the target gene from the human DNA, and at the same time plasmids made from DNA are isolated from a bacterial cell. The same specific enzymes are used to cut open the plasmid DNA. The open plasmids and the target gene are mixed together with different enzymes known as the DNA ligase, and this DNA ligase will join together the target gene and the DNA plasmid to form a recombinant plasmid. and the recombinant plasmids are inserted into the bacterial cells, and the bacterial cells are then allowed to multiply in a fermenter. The human gene can be switched on, and the bacterial cells containing the recombinant plasmid start to produce the human protein that we desire. So once bacteria have been genetically modified, as I've said, they can be grown and cultured in fermenters to produce large amounts of a desirable product. And the products that we have done this with so far include human insulin, enzymes for use with biological washing powders, enzymes used in the food industry, human growth hormone, and the cattle growth hormone known as BST. Also, we've been able to do this for human vaccines. For example, the hepatitis B antigen can be produced in this same manner. So just to look at these fermenters in more detail, how exactly would the industrial fermenter work? Well, you can see the diagram of an industrial fermenter in front of us here. The first thing to notice is that there is a steam inlet. This means that before and after use, the entire fermenter can be sterilized using high pressure and high temperature steam. The next line is the inlet. And at the start of the process, nutrients are fed in through pipes which can be opened and closed using valves. You'll notice that there are both temperature and pH probes. Now the temperature probe will be monitoring the temperature, and it will be working with the cold water inlets here and the cold water outlets. Fermentation produces heat, so to prevent the contents from overheating, the fermenter is surrounded by a water jacket through which cold water circulates, keeping the temperature at the optimum level for growth. Now, if the temperature begins to rise too much, we can add more cold water. If the temperature begins to drop, we add less cold water, so we can maintain that optimum temperature. The pH probe does a similar job. The pH of the contents can be adjusted by adding acid or alkalines to keep the pH constant. We have here some stirring paddles, and the stirring paddles mix up the contents, and this keeps the microorganisms in suspension in the liquid so that they get more exposure to the nutrients, and they help to keep the temperature even throughout the vessel. Some fermenters will use a jet of air to mix the contents instead of paddles. That's got the advantage of being able to add oxygen into the process so that aerobic respiration can take place. Now here is the air inlet, and as it says, if the organisms respire aerobically, filtered air is supplied through this inlet, and the air is filtered to prevent contamination but allow aerobic respiration. Now many microorganisms produce acidic waste products, so fermenters have to be made of materials which will not corrode, such as stainless steel or special alloys. After the transgenic bacteria containing the recombinant plasmids have been growing successfully in the fermenter for quite some time, we will then be able to open the harvesting outlet and allow the mixture to be released from the fermenter. This can then be purified so that the desired human protein can then be extracted from the mixture. It really is a fantastic way of being able to produce all of these human hormones in large numbers. Indeed, in the case of insulin, this now allows us to be able to treat diabetes type 1 and 2 successfully with insulin injections, because we can produce enough of this to satisfy the population that require them. It also means that there are no more ethical considerations when using either cow or pig insulin to treat diabetes. I hope you've enjoyed this short video on genetic engineering. If you have, comment below, let me know what you've learnt, and don't forget to like and subscribe for more videos from this channel. Thank you very much for watching.